I really didn't think that this would be possible, but the Baker Lab just designed proteins that grab hold of the floppy protein bits that most drugs can't catch. I'm talking about intrinsically disordered proteins, or IDPs, which are exactly what they sound like, relatively unstructured proteins, as opposed to the highly structured domains that we're perhaps more used to thinking about and more used to designing binders for. Typically, we think about protein binders fitting into a particular slot in a target, but if there's very little structure, then there are no slots to bind, making this a very challenging task. But it would be very useful to be able to reliably target IDPs. They're everywhere. Roughly a third of the human proteome contains a disordered part, and they're functionally important, with IDPs often driving protein association for better or for worse. So if you can change how those IDPs interact with each other, maybe you can break up harmful aggregates or otherwise modulate their organization. And Baker Lab does achieve this with their new binders. See in this example how this protein can't form clusters in the presence of the binder. So, how do you design a protein structure to target the unstructured? They take advantage of the fact that even disordered proteins adopt mini-structures at times, giving a binder something to latch onto. To design binders suited for those diverse IDP structures, they use a diffusion model, previously used for protein design, but here fine-tuned on relevant interfacing protein pairs. So all they give the model is the amino acid sequence of the target protein, and the model figures out an orientation of target and binder that looks like it would lead to strong binding. With that pairwise structure in place, another model back calculates which sequence would then best suit the binder. So per target, they generate a bunch of potential binder sequences and test a handful, and for all the targets mentioned in their paper, they end up with at least one strong binder. Really impressive. Of course, though, a big question is how specific are these binders? Because one could imagine that with such a floppy target, the binder would just bind to protein kind of broadly. They test each of their binders against a handful of non-target proteins to do a bit of specificity testing. However, I would love to see a more extensive specificity screen for these guys. Still, they're able to generate some really exciting binders. For example, one that binds to the disordered region of amylin, which is a protein that can form pancreatic aggregates and exacerbate symptoms in diabetes. The binder also contains a region that targets amylin to the trash can of the cell, leading it to be degraded and hopefully not aggregate. So it'll be really exciting to see what these kinds of binders enable in the protein aggregate disease space. Think neurodegenerative diseases or prions, and it's only a matter of time before the Baker Lab blows our minds again. So stay tuned.